Hello, this is my instructional demo on how to use the Ward's Natural Sciences model kits for Bi-12. We're going to start with the chemistry of amino acids and proteins, and I'll slowly work through the entire activity with you so you can see how the pieces fit together and what the beginning and the end of the molecules are. This is the general structure of your amino acid. Okay, take a look right here. Right, the blue is off the nitrogen. This is the unbonding site for the nitrogen. It's an unshared pair of electrons and not used during this lab. Okay, this right here is the unpaired carbon spot. This is the basics of an amino acid, but until you've actually filled that spot, it's not a true amino acid. Okay, when you're looking at the different groups, this one right here, okay, this is your amine group. Okay, and right here is your carboxylic acid. Okay, so now we're going to take a look at what we would bond to this site right here, okay, and how it changes it into the different amino acids. Here is our first amino acid. This is glycine. Okay, notice you still have the blank space on your nitrogen, but instead of having a blank space right here, you've now added a hydrogen. Okay. You still have your carboxylic acid part, and here is your amine right here. Okay. But now what you've done is you've added that hydrogen on that empty space in your central carbon. Okay, moving on. So now what you've done is you've added this radical group here. That's what we call that piece that fills that empty space. Okay, this is a radical group that is CH3. So one more carbon and 3 more hydrogens. Everything else stays the same, but now what we've got is alanine. And what you might be starting to notice is, oh, amino acids, right. Those are those 3-letter words that we use when we're making DNA strands. Okay, and we're coding through transcription and translation. Next, our next radical group here, and we're going to circle it. I'm going to try to circle it again. Okay, this is the radical group right here. In this case, you can see with the red there, that must be an oxygen and some hydrogens. Okay, so what we're adding there is CH2OH, and by doing that we've created serine. All the rest of the stuff is still the same. And lastly, the last group that you're asked to do is to add a sulfur group to the end. Okay, so again, that free radical right there, okay, is the sulfur group, and you're creating cysteine with that. And what you're adding is CH2SH. Okay, your carbon is right here, and your 2 hydrogens, there's one there and one there. And then you've got your sulfur, that's your sulfur, and your last hydrogen is right there. When you move on to step number 7, it talks about the peptide bond. So we've placed the glycerin model that we made in step 5 next to the alanine model made in step 6. Okay, So this is the glycerin here. And this is the alanine here. And then what it tells you to do is remove this section, okay, and this section, and then you'll start to bond them together. As you can see, I now removed those sections. I think the whole thing is upside down now, or backwards compared to the last screen, but you can tell the difference, hopefully. And now, when you look, what we can do is we've now bonded that OH with that hydrogen to create water, and then we've bonded the two molecules together, and then what we've done is had done what's called a dehydration synthesis and a peptide bond. So this is a peptide right here. This is water. And this is dehydration synthesis. Okay, so now we're going to start to form a polypeptide chain. We're starting with the serine from step 6, okay, and the dipeptide from step 7. Okay, so what's going to happen is you take off the OH, again from the carboxyl group, and you take off just an H from that amine group. When you take them off, you'll see on the next slide that they form water and a longer chain forms. Okay, so I've flipped the serine around, so what I took off was right here. Okay, and now I've joined them together. I didn't put the water on the slide, but that is where the new joint has happened. Okay, and now we've created this longer polypeptide chain. The next instructions. Okay, here's the large polypeptide that we just created in the last step, and here is the cytosine molecule that we created way back in step 6. Okay, following the same rules, we're going to take off the OH from the cytosine, this OH here, and then you take off one of the hydrogens off the end of the polypeptide of the amine group. Okay, when you do that, it leads to us ending up with an even longer polypeptide chain. Okay, so as you can see, they are all joined together now. I've now thrown the waters back in here. Okay, and the joints have happened, and now you have this long strand of amino acids. But this is only 4 amino acids strung together, so it looks pretty complicated, but in reality you need to pay attention that amino acid chains can be hundreds if not thousands of amino acids to create one protein. So this is our new protein. Okay, and it's a long strand.